Okay, so good afternoon, everybody, and welcome back. Our second class in microbiology. I'm just going to finish off a few of the things that I towards the end of our last um, presentation, and then we're going to carry on talking largely about cell structure. So, just to finish off that section, I want to remind you that the science of microbiology actually covers many different fields. Microbiology really refers to the study of anything which is not visible to the naked eye. So that encompasses many, many different things. And these are just a few of the areas. Bacteriology, which is largely going to be what we are going to be talking about. Bacteriology is specifically the study of bacteria. And bacteriology itself may be divided. Much of our discussion will be about clinical bacteriology, the study of interaction of humans and pathogenic bacteria. Mycology refers to the study of fungi in general. And the fungi are an incredibly diverse group. There are some pathogenic types of fungi as well. And so their clinical mycology would be the study of fungi. Parasitology refers specifically to the study of eukaryotic organisms. And although some of them, and in their adult form especially, may be quite large, if you think about the worms, intestinal worms, those sorts of things, you might wonder why a microbiologist is studying them. The answer is because they very often have stages of the life cycle, which are in fact microscopic. In addition to that, there are many, many, many eukaryote organisms which are microscopic, which are incredibly important parasites, incredibly important pathogens, such as malaria, sleeping sickness, all of those kind of things. All of those would come under the purview of a parasitologist. Now, here's a very important field because we are going to be talking at some length eventually about viruses. And of course, for us at the moment with the current COVID epidemic, it should be familiar to you that there is an entire science of virology, the study of viruses. And viruses are obligate pathogens. They are obligate parasites. They have no other way of existing or of reproducing. So the existence of viruses has been known for a very long time. In fact, they were first really described in 1892 by Ivanovsky, a Russian scientist, but their presence had been noted but not explained prior to that. The way in which microbiologists discovered viruses is in itself quite interesting. Many media that we grow bacteria on are extremely sensitive, and they break down if they're heated. So they're very difficult to sterilize. And early on in microbiology, it was discovered that you could sterilize fluids by passing them through a very fine filter. And the filter filters out all the bacteria and filters out bacterial spores. And ideally, what you end up with is a sterile liquid coming through on the other side of the filter. We still do this in microbiology if we're using sensitive media. However, microbiologists noted that in fact, when they passed liquids through these filters, which they were sure were taking out all of the bacteria, the fluids that came through could, under certain circumstances, still be infectious. And Ivanovsky noticed, in particular, that this was true when he was working with plants, and he worked especially with tobacco. And he was working on tobacco diseases and initially thought that the diseases he was seeing were caused by bacteria. But he found that when he made solutions of the plants, the infected plants, and passed those liquids through these filters, which should have taken out all the bacteria. The fluid still remained infectious. And he proposed that, in fact, there were infectious particles passing through the filter. Initially, he thought that they would be functioning much like bacteria. Of course, they don't. They function in a totally different way. But nonetheless, he showed that filtered material like that was infectious to tobacco plants. But it remained until 1935 before we actually saw those infectious particles. And that was done by Wendell Stanley. And he was the first to visualize viruses and using what was then a brand new piece of equipment, which was the electron microscope, which viruses are far too small to see under a microscope. They're far below the ability of a microscope to resolve, but they can be visualized using an electron microscope, which can visualize things which are thousands and thousands of times smaller than a bacterium. Here's one of the earlier pictures of tobacco, of this virus, particular virus, and it's called tobacco mosaic virus, a very, very famous virus because it was the first one to be clearly elucidated first of all by Ivanovsky and then visualized by Stanley. Here, as you can see, it's a long filament like this, and we'll see it again when we talk about viruses later on in the semester. Okay, so here's now in our modern times, here is the huge field of endeavor in microbiology. And that is in the study of molecular genetics. And many people would call this the fourth golden age of microbiology. And it is the study of microbial genetics. And it depends upon the fact that DNA structure in the mid-20th century was just finally described by Watson, Crick, and Wilkins. And the structure of DNA was such that it immediately became obvious that how genes actually functioned and how DNA replicates and all the rest of it. And this led to this huge field of study called molecular biology, which is the study of how DNA directs the cellular processes, especially through the synthesis of protein. And the study of genomics is the study of all of an organism's genes. And this has provided us with ways not only of classifying organisms, but of displaying relationships between them, displaying ancestry in evolution of organisms, and also has opened up this whole field of what we refer to as recombinant DNA technology. And it depends upon the fact that, as I told you before, all living organisms use basically the same mechanism. And that is the DNA is transcribed to messenger RNA, and messenger RNA is translated to protein. All organisms use that system. And this opens up the possibility that we can begin to take genes from one organism and put them into another organism and get that organism to produce the proteins from the original gene. This has become extremely, extremely useful. And the reason for that is that many products can be produced by bacteria whose genomes have been altered, whose genomes have had useful genes inserted into them for specific products that we are interested in. And bacteria are very easy to grow. They're very easy to grow in bulk, in very, very large quantities, hundreds of thousands of gallons, if need be, of a particular bacterium producing a particular product from an inserted gene can enable us to produce vast quantities of useful substances. And this is the science of recombinant DNA, and very currently, industrially and scientifically, a very, very important field of endeavor. So in our class, of course, we are going to concentrate on microbial human interactions, and we're going to talk especially with some emphasis on pathogenic bacteria, but we will also have reference to the fact that we have, as human beings, we have a huge population of very, very useful, friendly bacteria coating our skin, coating the insides of our gut, especially. And that whole area of study is called the microbiome. And it's the study of the interaction of human beings with their normal, naturally occurring, non-pathogenic bacteria, beneficial bacteria in actual fact. In addition, unfortunately, in this class, we don't have much time. We only refer to it in passing, really. The fact that the functioning of our entire global ecosystems depend absolutely upon the functioning of microbes, especially to recycle elements. On our planet, we only have a finite number of molecules. We do not have material continually arriving, a very small amount from space. But basically, we're dealing on our planet with the original molecules that we ended up with forming the planet right at its very founding about 4 billion years ago. Those elements have got to be recycled continuously. They have to be reused. If they were to get bound up into materials which did not decay, we would eventually run out of molecules. But thanks to the action of microbes, especially bacteria and fungi, very complex organic molecules are broken down continuously as organisms excrete, as organisms die. Their molecules are broken down and are recycled to the global ecosystem. And that is the essential interaction of naturally occurring microbes in the environment. And that relationship, the study of the relationship between microorganisms and their environment including other living things, is the field of study of microbial ecology. Unfortunately, we don't discuss it in any great detail. But all of the elements, carbon, oxygen, nitrogen, sulfur, all of these things are continually built into biological molecules, the processes of metabolism, and then have to be broken down to be recycled to other living things. That is done by microbes. So I'm going to repeat these distinctions several times. Let's move on to actually discussing the structure of microbes, the cellular structure and function of microbes. Remembering, we've already said that we recognize two broad categories of cell, of type of cell on the planet. The first type of cell is the one which we are going to be most concerned with in this class, and those are the prokaryotic cells. There are two groups of organisms which are prokaryotic, and they are the bacteria, and another group which we won't spend much time talking about, called the archaea. So the bacteria and the archaea together, those two groups comprise the prokaryotes. Prokaryote actually means before a nucleus. You can also think of it as being before a true cell, but that's how it was originally thought of. Bacteria and archaea are very much truly a cell. So the primary fact about the prokaryotes is that The only membrane which they possess is the cell membrane, the plasma membrane, which encloses the cytoplasm. They do not have any membrane-bound bodies or organelles inside the cytoplasm. So obviously there is no nucleus, because the nucleus is a membrane-bound organelle in the eukaryotes. In general, the bacteria, the prokaryotes rather, are very much smaller than the eukaryotes. And there's an important reason for that, and that is that they become limited because all of their function takes place inside the cytoplasm. If they get too large, molecules diffuse away from one another, and the important biological reactions are difficult. In eukaryotes, special functions are contained within their own membrane, so the molecules are close together. So the prokaryotes are restricted in size. They are generally smaller. There are some large bacteria in actual fact, but they are specialized. But in general, the prokaryotes are much smaller than the eukaryotes. They have their genetic material is, of course, DNA, the same as every other living thing. But they only have a single chromosome, basically. And the chromosome is in the form of a circle. That is their principal type of genetic material, a circular chromosome. They reproduce predominantly by asexual reproduction. There are a few special instances which you will hear about where bacteria can exchange DNA, but it's not sexual reproduction the way that we think about it. In addition to this large chromosome that they have, which is their principal genetic store of information, bacteria may also have small amounts of extra chromosomal DNA, DNA which lies outside the principal DNA of the chromosome. And these are very small circles of DNA called the plasmids. And the plasmids are tremendously important, if for many reasons. The principal reason being they carry often very useful information for the bacterium. They may carry information, for example, which allows the bacterium to be resistant to antibiotics, that sort of thing. They may also carry virulence factors, things which, genetic information which enable the bacterium to become a better pathogen. The second really important thing about plasmids is they can be handed on from one bacterium to another under different circumstances. And they can, in fact, be picked up from the environment. If a bacterium which has plasmids, for example, dies and bursts, it can release its plasmids into the environment. And those plasmids can then be picked up by other bacteria. So bacteria can acquire characteristics which they did not originally have. The plasmids are fundamentally infectious. They can be passed from one bacterium to another. It's a way of transmitting genetic information. So the eukaryotes, we don't actually in this class spend a huge amount of time talking about eukaryotes. I will periodically refer to the eukaryote microbes in one way or another, but most of our interest is in clinical bacteriology. But we will spend some time talking about the structure of the eukaryote cell, because it's so important for us to understand the difference between prokaryotes and eukaryotes. The eukaryotes, again, they have a cell membrane, a plasma membrane around the cytoplasm. All living things do. But in addition to that, they have areas of the cytoplasm that are themselves enclosed in membranes so that special functions can be undertaken by those. Those areas which are enclosed by their own membranes, we refer to as the organelles. And the most obvious organelle of all is the nucleus of the eukaryotes. And the nucleus of the eukaryotes contains the vast proportion of the DNA of the organism. And that often forms an easily visualized organelle, which we refer to as the nucleus. Because of this organization, because they have special functions isolated to these organelles, the eukaryote cell is often very much larger than the prokaryote cell. And the cell just looks and very much more complex. It's the cellular functions inside the cytoplasm are very much more complicated than they are in the prokaryotes. In addition, the DNA inside the nucleus is organized differently to that in the prokaryotes. In prokaryotes, it's a circle. It's a circular chromosome, and it's a single chromosome, fundamentally. In the eukaryotes, the DNA strands are linear, and they are organized into separate chromosomes, and there's usually a number of them, a number of these strands of DNA, not circular, but in a single strand, each of them. So the eukaryotes usually have more than one chromosome in the nucleus. In the eukaryotes, I should also mention, just so that it's clear at the very start, the vast proportion of DNA is inside the nucleus, but there are one or two other organelles inside eukaryotes, which may have a small amount of DNA. The most important of these are the mitochondria, which all eukaryote cells have unless they're very, very specialized. And the chloroplast in the plants. Mitochondria and chloroplasts have their own small amount of DNA. Okay, so here let's have we'll think about some of the types of microorganisms which we will discuss, and I'm going to just discuss these briefly now and give you some of the characteristics of these different groups. So we're first of all, of course, our major interest is in the prokaryotic groups, and these first of all the bacteria. Bacteria are fundamentally single-celled organisms, prokaryotic. They have no membrane-bound organelles, nothing like that, and they divide, they grow, and they will reproduce by binary fission. That is, the cell grows to a certain size. All of the DNA inside the cell is duplicated, and the cell divides into two essentially identical, usually smaller cells, which then themselves grow to a certain size and divide again. We say that they are single-celled, but they may associate with one another. After they have divided, they may remain associated with one another. And in addition, depending on circumstances, they will actually deliberately associate with one another. Even genetically distinct bacteria of the same kind, and sometimes of different kinds, will deliberately associate with one another in particular ways that we could recognize. A fundamental distinguishing feature of the bacteria is that they have a cell membrane, of course, all living things do, but around the outside of the cell membrane, they usually have a cell wall. And the distinction between the two is very important. The cell membrane is a living entity. It is a living, functioning part of the cell. The cell wall around the outside is an inert construction. It has no living properties. It is very important. It has some very important functions to perform, as I'll describe in a while. but it is completely distinct from the cell membrane. They are two different things entirely. The cell membrane around the cytoplasm is a living, functioning part of the cell. The cell wall around the outside is secreted by the cell for particular purposes that we'll discuss in a minute. The other distinguishing feature of bacteria, which they do share with a couple of other groups, as we'll hear in a minute, is that the bacteria are unable to swallow things. They are unable to take in material, particulate material from the outside and digest it or whatever inside the cytoplasm. Instead, they acquire all of their nutrition and everything by diffusion. And as a result, what they need to do is when they, in their environment, they need to break down molecules in their environment till they are sufficiently small that they can diffuse into the cell. So the way in which bacteria function is to release enzymes into their environment. Those enzymes digest extracellularly. And once molecules have been broken down small enough to be absorbed by the bacterial cell, they can move into the cytoplasm by diffusion. Often, however, there are mechanisms to ensure that these molecules do make their way into the cytoplasm. As we'll see in a while, the cell membrane is very good at allowing particular molecules to get through to the cytoplasm. Some bacteria, that is referred to as a heterotrophic function of bacteria. Heterotrophy means feeding on molecules which have already been created. Okay, those molecules that exist outside them, those molecules already exist. They just need to break them down into small enough parts that they can then absorb them. But very, very, very importantly, there are bacteria which are autotrophic. That is, they can take carbon dioxide and they can take water, these two simple molecules, and they can bind them together using energy, usually energy from the sun, but occasionally energy from other sources. They bind them together and they manufacture their own organic molecules. So that is the most important way in which this is done, is through the process of photosynthesis. So here, by the way, up here are bacteria seen under a microscope. They are stained a particular color. They're not naturally this dark blue color. But if you look very, very carefully at this, you'll see that these are actually all rods-shaped bacteria all joined together into these web-like structures. These ones down here are different. These long threads here are, each of these is an individual cell here, and then each cell joined to another cell to form these long filaments. This is a group of bacteria called the cyanobacteria. And the cyanobacteria are just hugely significant in evolutionary terms, because it was in the cyanobacteria that the process of photosynthesis actually evolved. About 3.5 billion years ago, this process of photosynthesis evolved in this particular group, very, very successful group of bacteria, the cyanobacteria. And cyanobacteria, in actual fact, provide nearly 40% of our oxygen in the atmosphere that we breathe. The rest comes from plants and that sort of thing. But the process itself evolved in bacteria. So, and we still derive a huge amount of our breathable oxygen from the cyanobacteria. So this is the other prokaryote group. We're not going to talk about them very much, although they are extremely interesting. Their interactions with human beings are minimal. There are no pathogens. These are the archaea, a very strange group, but of enormous interest to evolutionists, because We actually believe that the eukaryote cells evolved from archaeal cells. Our eukaryote cells are more aligned with archaeal cells than with the true bacteria. They are still prokaryotes, and they have many of the prokaryote characteristics, but they don't have peptidoglycan cell walls. Now, this substance, peptidoglycan, this is our first hearing of it. First of all, here, the bacteria have a peptidoglycan cell wall. The archaea do not. And you will hear a lot more about peptidoglycan in a minute. The archaea live often in very, very strange circumstances. We are often, many of them we refer to as being extremophiles. And an extremophile is something which lives in a very harsh environment. The archaea are able to live in environments, for example, where there's very, very high salt content, or where the pH is very low, where it's an acid environment, or very, very alkaline environment. They also perform many strange metabolic processes. So, for example, most of our methane on the planet is produced by the archaea, and it's produced by them in metabolic processes and nutritional and metabolic processes. And they, for example, occur in cow stomachs, in cow rumens. That's why cows belch methane the whole time. So this is they're a very important group, but unfortunately, we don't have time to discuss them in any great detail. If you're interested, there's an abundance of literature that you can follow up. But remember them, the principal reason to remember them, first of all, they're extremophiles, produce methane, that sort of thing, do some interesting things, but they are our other prokaryote group. So now let's talk a little bit about some of the eukaryotes. And the first group to talk about are the fungi. Fungi, in fact, true eukaryotes. They have very complex eukaryotic cells, membrane-bound organelles, and especially membrane-bound nucleus. They reproduce asexually and sexually as well. In other words, they actually exchange DNA information in order to reproduce. So they are true eukaryotes. They also have cell walls like the bacteria, but the cell wall in the fungus is not made of peptidoglycan. Instead, it's made of another polysaccharide called chitin. And chitin you may have heard of, because chitin is the substance which, for example, the arthropods, insects, those sorts of things, they use chitin as well to form an exoskeleton. In the fungi, it's used as a cell wall. I'll discuss the function of cell walls in a little while. They share one important characteristic, however, with the bacteria, and that is that they digest, they are heterotrophic, entirely heterotrophic, and they they release enzymes into the environment, break down complex molecules in the environment, and absorb the products. So in that way, they are very like the bacteria. And it's for this reason that the bacteria and the fungi are so important in recycling of elements, of recycling of compounds, because they go in the environment, they will break down very, very complex biological molecules into simple compounds, which they themselves can then absorb. And very often, for example, in the case of carbon, they will break down those compounds for energy and will release the carbon as carbon dioxide. So there are two broad kind of morphologies, if you like, for the fungi. The first are complex bodies made of cells associated with one another. You can think of all the way from these kind of fungi on bread and everything, all the way to the mushrooms and those sorts of things that are easily visible. So these are complex units, are made up of many cells, very carefully organized into a particular morphology. But there are also many fungi which are unicellular. And these are of particular importance in health, in human health and animal health as well, because these are the yeasts. A unicellular fungus is referred to as a yeast. Most yeasts are non-pathogenic, of course, but there are several yeasts which are pathogenic, both to humans and animals. The other reason we are particularly interested in the fungi is because many of the fungi produce antibiotics. They produce compounds which will actually kill bacteria and other living things as well. And you need to think about why. Why is it that a fungus would produce an antibiotic to kill other living things? Well, there could be a number of reasons, but the most important one is this. In these environments, such as, just think of a forest floor or a decaying animal or something like that, there are a gazillion organisms trying to monopolize that food source. There are a huge number of different species become involved. And what they are doing is they're competing with one another. They're competing for space, just space to grow. And they're competing for the nutrients and everything in the environment. And any advantage which an organism can gain over other organisms is going to benefit them. And one of the things which many of them do, not only fungi, but bacteria as well, is they begin to release compounds which kill their competitors. So they free up space and they free areas for colonization because they release these compounds which will kill their competitors. Fungi, in particular, compete against bacteria. So many fungi will release into their environment, will release antibiotics which kill bacteria. We have discovered how to culture those fungi and extract those compounds to use clinically to kill bacteria. So that is the origin of many of the clinical antibiotics which we use. Quite apart from that, fungi are extremely useful to us in many other ways. We eat them. There are many edible fungi. They impart very distinct flavors to things that we do like, such as cheese, pickles, those sorts of things. So the fungi are very diverse. Some of them extremely useful. Some of them very much a part of our microbiome, and in the environment extremely important because of their recycling ability. So there's the next group of eukaryotes to mention are those which are largely unicellular eukaryotes. Now, these used to be known as the protozoa. That's why the name here is put into inverted commas. It's in inverted commas because this is not a term which is really recognized as being valid anymore. Instead, the protozoa belong in a group called the protists. And the protists, I mentioned before, very poor grouping, but they are the organisms which are neither plants, nor fungi, nor animals. Anything which is not a plant, a fungus, or an animal gets thrown into this group called the protists. And included in that is the whole group of eukaryotes, which we used to refer to as the protozoa. And they are just so diverse that you realize that this is a very poor grouping. There's not a lot that unites the different members of the protists or the protozoa. Some of them are heterotrophic. Many of them are heterotrophic. Some of them are autotrophic. This is a very famous one called Volvox. And this, if you may at some time in your career see this in lab. It is an aquatic, photosynthetic. Many of them are in fact photosynthetic. Here is one that we will hear about later on in our class. This is obviously not photosynthetic. This is called a trypanosome. This is a single-celled organism. Here's one cell here. There's another one here. This is in a blood smear. And this is a blood-borne disease called trypanosomiasis, or sleeping sickness. And this is caused by one of these protozoan protists. I put these two together because, in fact, these, believe it or not, when you look carefully at them, you discover that these trypanosomes are actually closely related to a free-living protozoan called Euglena, which is a photosynthetic organism. It's very, very strange, but it is in fact true. These protozoa sometimes look incredibly diverse from one another. Then when we look at their molecular biology, we discover that they're actually closely related. Okay, so also in the protists, and closely related, for example, to that organism that I just showed you here, Volvox, are the algae. The algae of these protists, their individual cells are photosynthetic, etc., but they are multicellular. And in fact, some of the largest organisms on Earth are algae, and in particular, the brown kelps, which occur on the west coasts of Africa, Australia, and of California and South America. These are, in fact, some of the largest organisms on the planet, believe it or not. But the algae have a characteristic. They also have a cell wall, but their cell wall is fundamentally constructed with cellulose, although it may have some other compounds embedded into it. In this, they share many characteristics with the plants, but they are very distinct from the plants. They are far more primitive, far simpler. The algae are found throughout aquatic systems, and they're also even found in soil, in moist and damp soil. They are tremendously important photosynthesizers and also contribute a significant amount of oxygen to the breathable air. So those are the only kind of, by the way, where we include, formally, we would include study of algae in microbiology because they have microscopic stages of the life cycle. Okay. There are one or two algae, in actual fact, which are pathogenic. It's very rare for them to be pathogenic, but there are one or two kinds which rarely cause some problems. But we, certainly in microbial ecology, in the study of microbial ecology, the study of the algae is extremely important. Okay, so now we'll leave those few eukaryote groups behind, and let's just talk a little bit about the viruses. As we've said, the study of viruses is virology. And virology is the study of viruses and their interaction with their hosts. And the viruses are all obligate parasites. There is no free-living virus. There's no way for a virus to live, to actually function or complete a life cycle or reproduce as a free living system, because they are, first of all, there's no cell structure. They don't have a true cell. The second thing is, of course, they have no metabolism. outside of their parasitic condition, when they have infected a cell, they are totally unable to do anything. There are no life processes going on in a viral particle when you look at it. So here are some viruses here. As you can see, we'll learn a lot about their structure later on. But this is a relatively simple virus, and it has a membrane around the outside here, there. And then on the inside here, it has something containing the genetic material. Remember that our definition of life said that all life is based on a cell. Viruses are not cellular. Second of all life is based on DNA, and on the DNA being transcribed to messenger RNA and producing protein through translation. That doesn't occur in the viruses. Their genetic material is, in fact, not necessarily even DNA. Depends on the type of virus. The COVID-19 virus that we're dealing with at the moment, for example, is an RNA virus. It doesn't have DNA at all. It has RNA as its genetic material. But there are other viruses which do have DNA as their genetic material. But inside the virus, if we were looking at this, there is nothing happening in here. There is no metabolism. The DNA or whatever is not being transcribed or translated or anything. It's just sitting there waiting to be injected into a host. So at their simplest, viruses consist of a piece of genetic material surrounded by a simple protein coat. That can be elaborated on a little bit. And so, for example, in the COVID-19 virus, around the outside of the virus, there is another membrane, which is derived from host membranes. But that is not always depending on the type. That depends on the type of virus that you're talking about. The really important thing about the viruses is that they are inert outside the host, but once they infect a host, what they do is they change the genetics of the host. They actually inject their genetic material into the host, and that genetic material conscripts all of the functions of the host cell to produce more viral particles. And those viral particles are then expressed from the cell in one way or another to go and infect other cells. So the virus is completely inert as long as it's outside the host. Once it's inside the host, what it's doing is it's changing the host cell into a viral factory in one way or another. The last group that we will only briefly refer to, and you may see in lab, are the multicellular animal parasites. And these are also part of microbiological studies because although adults may be quite large, very, very often they have microscopic larval stages, or they may themselves be so small that they are in fact microscopic. But these are multicellular animals, often very, very, very highly adapted to living as parasites. The biggest group that we spend time considering are parasitic flatworms, like the tapeworms, that sort of thing, and the parasitic roundworms, things like that. And these worm-like things we refer to as helminths. You'll often see, for example, that somebody is giving an animal or has been told themselves, their children, to take an anti-helminthic. This is a treatment for worms, for the presence of worms in the body. Very often, in actual fact, the adult worms cause less problems than the larval worms do. This is particularly true of things like the roundworms that infest our gut. Most of the problems that the roundworms cause are in their larval stages, which migrate all over the body before they end up being swallowed and settle finally in the gut. So that would be a province of probably of a parasitologist to examine that. So we've already come to the conclusion, as you've listened to me rabbiting on, that we're looking at an enormously diverse group of organisms. In fact, there are probably tens of thousands of species of named bacteria, or recognized bacteria rather, not necessarily even named yet. And the vast majority of prokaryotes we know virtually nothing about. They are just hugely diverse. But when an organism is recognized scientifically as being distinct and has been characterized so that it can be recognized again, it is very often given a Latin name. And that Latin name is very important because it enables it. In fact, the name is like a code. The name is like a code for everything that the organism does and everything that characterizes the organism and that makes it different to other things. This is not a simple matter. In the very old days, it was recognized that we wanted to distinguish one living organism from another. And what they used to do was they used to write up a lengthy description in Latin which applied to that particular organism. So a rose would be described as, a flower with thorns, with leaves and thorns that produce a sweet smelling blossom, et cetera, et cetera, et cetera, et cetera. This was very cumbersome, very, very cumbersome to have to compare these in order to distinguish one thing from another. It's just have to compare these lengthy descriptions one to the other, often the descriptions not particularly accurate. This gentleman, Carolus Linnaeus, that's his Latin name. He is actually a Swede, and his last name was Linne, L-I-N-N-E, Carl Linne. And he did something very important in science, and that was he established a standard way of naming organisms. And his innovation was to describe organisms just with two words. The first word is called the genus name. The second word is the specific epithet, the specific name. So he said all living things can be described with these two, a genus name and a specific name. And once a scientist recognizes that an organism is entirely distinct from other organisms, they can then describe that organism, but they must assign to it this code, this, first of all, the genus name, and then the specific name. And very usefully, those names often are somewhat descriptive. So this is how all living things are named. I will later on explain to you that it's a little bit difficult sometimes with bacteria. But for much of the history of bacteriology, of microbiology, prokaryotes have been named in the same way. Once they have been recognized as being absolutely distinct with certain characteristics, they are assigned a genus name and a specific name. And so here are some rules which you must know about this naming, this system of nomenclature. The scientific names are always italicized when they're printed. And if you can't italicize them, and if you're writing them out by hand or whatever, you underline them. That underlining is an instruction to a printer. In actual fact, if a printer sees that and renders your writing as a printed article, they will put anything underlined into italics. So scientific names, both the genus and the specific name, are italicized. The genus name is capitalized. The specific name is not. It is all lowercase. And they are Latin names. There's a little bit of latitude here because they are Latinized often. And they'll take a word and they'll render it as Latin, and sometimes there's a lot of leeway as to how you do that. You'll actually discover that some things are, the name is fundamentally Greek, but it's been Latinized, for example. Most usefully, the names are descriptive. They tell you something about the organism, but it's not necessarily so. And sometimes, as you probably know, especially the specific name, but its genus name as well, may refer to a scientist in honor of a scientist maybe who discovered it originally or something like that. So here's a good example, and this is one that you are going to encounter over and over and over again, not only in this class, but also in lab. And this is the famous, most commonly cultured bacterium in the lab, Escherichia coli. So here is the genus name there. There are other species of Escherichia. So Escherichia coli is a particular species, and it has particular characteristics which have been described so that we can always recognize Escherichia coli and distinguish it. The genus has particular characteristics. The species has particular characteristics. Together, they describe one particular kind of organism. This is not to say that there is not variability in that organism. There is. But as long as they satisfy the original descriptions, they will be placed here into the genus Escherichia and the species coli. Escherichia is actually a name which honors their original discoverer, Theodore Escherich. And so Escherichia is after somebody, a name for somebody. But coli describes where they are principally found, and that is in the gut. That's very useful to know, because this means that this tells us something about this organism. It describes the habitat where they principally occur. They may occur outside. You may be able sometimes to recover them from the environment, but that is usually an indication that there is fecal contamination, that this is originated. They survive outside the gut, but it's usually a sign that there's been contamination of water. So that's Escherichia coli. That's its full scientific name. Here are some others that you need to know and that you will hear about. Now, this name here, Staphylococcus aureus. Staphylococcus aureus, this whole name is descriptive. First of all, this here, coccus, we're going to hear in a while, that describes a spherical shaped bacterium. So that is a coccus. So we know automatically. We look at this name, we know it's a coccus. Staphylo, this is a Latin term, and it means in a bunch. It actually refers to grapes. It means that they're in a bunch like this. And that's very descriptive. When you see a picture of Staphylococcus, you'll realize why. Staphylococcus, cocky, in a bunch. Aureus means golden. And that is because when you grow the Staphylococcus on certain media, it turns a golden yellow color, which is very characteristic. So the Staphylo, clustered, coccus, spherical, aureus, gold colored. On Staphylococcus aureus, there are many species of Staphylococcus have been recognized, but there's only one Staphylococcus aureus. There's only one species which conforms to all of the characteristics of Staphylococcus aureus. Here's another important one. Here's the genus Neisseria. Now, this is also named, by the way, for somebody, Neisseria. But here we'll think about two species of the same genus. Neisseria meningitidis is the one, Neisseria gonorrhea is the other. It's very easy to recognize the genus Neisseria, especially when you look at it under the microscope, because this is a coccus which occurs in twos. Principally, it occurs as two cocci joined together like this. We call that a diplococcus. But very unusually for the cocci, this diplococcus is gram negative. You'll hear in a while what we mean by gram. It is a particular stain which tells us about the nature of the cell wall of the bacterium. And Neisseria is one of the very, very few gram negative cocci. Most cocci are gram positive. So this is a gram negative diplococcus. We know immediately we need to think about Neisseria as soon as we see a gram negative diplococcus. Both of these species look very, very similar, but they do completely different things. Neisseria meningitidis can cause a very serious infection of the brain and the spinal cord called meningitis. That's where it gets its name from. Neisseria meningitidis, able to cause meningitis. So that is one kind of Neisseria. The other Neisseria is Neisseria gonorrhea. And Neisseria gonorrhea looks almost identical under the microscope, but it causes completely different disease, which is a sexually transmitted disease of gonorrhea. So they behave completely differently. They look much the same. They are in the same genus, but they are distinguished by their specific names, which indicates to us that they behave completely differently. So sometimes when you're writing a paper or whatever about a restricted number of bacteria, it's a pain to have to repeat the whole long thing all over again. So here's the rule. First of all, we're going to write a little paper, and we're going to compare Escherichia coli and Staphylococcus aureus. So our very first sentence will be Escherichia coli written out in full, and Staphylococcus aureus are found in the human body. We write them out incomplete the first time that you use the names. But the second time and subsequently that you use the name, you can abbreviate the genus name. So now, subsequently in your writing, you can say E. coli. So E. coli, Escherichia coli, Staphylococcus aureus are found in the human body. E. coli is found in the large intestine, and S. aureus is on skin. And by the way, here they are. This is Escherichia coli, this rod shape here. Its shape actually varies quite a bit depending on how you grow it. But this is a classic shape. There's a long rod. Here is Staphylococcus. Why Staphylococcus? Coccus because it's spherical. Staphylo because it occurs in bunches. There. So that's where it gets its name from. All right. So let's, I tell you what, just stop your recording here and just wait, okay? Just go do something for 5 minutes. Just clear your head for 5 minutes and then come back to the recording. But take a break now, and we will continue in a second. Okay, so welcome back. First, we'll have a good stretch. Stretch out, shake yourselves out, and let's finish what we're talking about for today. This, by the way, this PowerPoint set will last for a couple of classes, okay? You don't think we're going to get through all of this in one day. We won't. So let's just remind ourselves again about the basic structure of prokaryote cell before we start talking about some of the details. First of all, remember, the prokaryotes, of course, lack a nucleus, and they lack all of the membrane-bound internal organelles that are characteristic of the eukaryote cells. They share with the eukaryote cells the following characteristics. They all have a cell membrane around the outside, as all living things do. They have, within the cytoplasm, they have most of their DNA organized as a chromosome, although in the prokaryotes, it is a circular chromosome, a single circular chromosome, maybe some small plasmids. So they have DNA driving all the cell processes and then interacting to drive those cell processes. In common with the eukaryotes, they have these small bodies called the ribosomes, which are responsible for the production of protein from the recipe messages contained originally in the DNA. So they share with eukaryotes DNA, a cytoplasm, ribosomes, and the cell membrane. Now, in the bacteria, almost all bacteria around the outside have a cell wall that we're going to be hearing quite a lot more about. So outside the cell membrane, there is a cell wall. Many bacteria have lots of other things associated with them, as you will hear. They may have flagella on the outside. They may have protein extensions. They may have slime layers, all sorts of things we will talk about. They're not necessarily just the simple envelope structure that is shown here. Nonetheless, they in general are much smaller, much less complex rather, than the eukaryote cells. And we've already heard about two broad groupings, the bacteria, the heterotrophic bacteria, and the cyanobacteria, which are autotrophic bacteria. So here is a typical bacterial cell. And this could be any one of a number of cells that you may see in lab. And first of all, just look at its general structure. And we'll talk about some of these other outer structures in a while. So here's the cell membrane, often also called the plasma membrane. And here's the DNA on the inside of the cytoplasm, which lies in that area enclosed by the cell membrane. The cytoplasm is also full of ribosomes. Most of the DNA of the cell is organized as this very long, compared to the size of the cell, this very long circular chromosome. Here it is here. It is not randomly just lying around inside the cytoplasm. It is very, very carefully organized. It's organized into a dense area of DNA, which we call the nucleoid. Sometimes in very large bacterial cells, this is even visible with a very good microscope, can be visualized, but is easily recognized on an electron microscope because we can stain for particular molecules. And here it is here. This is the DNA all organized into this particular area. It is called the nucleoid. In a eukaryote cell, it's the nucleus because it has a membrane around it. But in the bacterium, in the prokaryotes, it has no membrane around it. So it's just referred to as the nucleoid. In addition, there may be additional pieces of DNA in the form of the plasmids in the cytoplasm. Cytoplasm is full of ribosomes, just as it is in the eukaryotes. And the ribosomes are producing protein. And they're producing protein under instruction of messenger RNA, which is produced from the DNA code for the protein. So there's a cell membrane. Around outside of the cell membrane is a cell wall. And we'll discuss the function. There are a couple of functions for the cell wall. The most obvious one is that it's produced in such a way that it actually gives structure to the cell. And the characteristics, shapes of the cell that we see are imparted by that cell wall. So it is produced in a very particular way. So it adopts a certain shape that the cell conforms to. And it does so repeatedly, right? As the cell divides, every time it divides, it produces a similar looking cell, which constructs a similar cell wall. All that, obviously, is encoded. It's genetically determined what shape the cell is. And we'll see some of the cell shapes in a minute. Outside the cell wall, you may also have all sorts of other structures. There are hairy kind of extensions and long tubular extensions, and sometimes slime layers and a capsule and all sorts of things that we'll hear about in a while. So outside the cell membrane, there is this whole envelope structure, the cell wall, and then all of the associated things with it. One of the most noticeable characteristics often associated with the bacterial cell is the fact that they have flagella. These long extensions, very complex things, very complex structures. And I'll show you again in a while how they actually work. And they are strongly associated with the cell membrane, with the plasma membrane of the cell. They are actually embedded in it. And there are various types of flagella. Well, I wouldn't say types of flagella, but there's various ways in which the flagella can be arranged. Not all cells have flagella, and not all cells have flagella all the time. Sometimes they develop flagella for particular stages of growth and distribution, everything, and then lose them. Some bacteria are never flagellated. There are no, for example, there are no true cocci which are flagellated. It's always a feature of rod-shaped bacteria. So these are flagella, and they are responsible for moving the bacterium through the medium that it is growing in. That's the principal and the most efficient form of bacterial movement. But in fact, bacteria can move in other ways as well. They have a form of movement against a surface, which is called gliding motility. They also have a strange twitching kind of motility. And both of those, both gliding and twitching motility, are imparted by these extensions around the outside here, which are called the fimbriae, around the outside of the cell. We'll hear about several of these again in a minute, so don't panic for the moment. In addition to that, inside the cytoplasm, there may be many structures. They are not membrane-bound structures. They're not organelles, but there may be things, for instance, areas of the cytoplasm which are used for storage, sometimes for lipid, other crystalline substances, all sorts of things we'll hear about in a second. So there may be other things included inside the cytoplasm, but none of them are membrane bound. The only thing that I will mention is that in the case of photosynthetic bacteria, the photosynthesis of those bacteria takes place on the membrane. Many metabolic processes are associated with the membrane. In the case of photosynthetic prokaryotes, very often what they will do, they need a huge surface area of membrane to perform photosynthesis efficiently. So what they will do is they'll take the cell membrane and it becomes enormously long and folded inside, but it's still cell membrane. It's not a separate cell. It's not a separate organelle enclosed by membrane. Okay, so I've already said the shape of the cell is something which is genetically determined. and it is reproducible. If we grow bacteria, they will grow repeatedly. Every time they divide, they'll produce a similar kind of shape. Not necessarily identical, but very, very similar. So let's have a look at the different types and different kinds of cell and the way in which they are organized. So first of all, this is already familiar to you. This is a rod-shaped bacteria, and a rod-shaped bacterium is also referred to as a bacillus. So a bacillus is a bacterium which is rod-shaped. It's a little bit confusing because there's actually a genus called Bacillus as well. So you will encounter a genus called Bacillus in lab, for example. Obviously indicates that it's rod-shaped. But we also, so this is E. coli. So we say E. coli is actually a bacillus with a little b. It's a bacillus. It's a rod shape. I'm going to just jump over here. This is, you've already seen. This is a coccus, a spherical, or more or less spherical. Sometimes they can be a little bit squished. Sometimes you need to use a bit of judgment as to whether it's bacillus or just a short, very fat rod. But these are very clearly spherical. And this is Staphylococcus. Staphylococcus easily recognized because it's in bunches. But here is another coccus. And this is Streptococcus. And Streptococcus is in chains. And that's what it means. Strepto means straight in Latin. So these are Streptococci. The cells are joined together in chains, not in bunches, but in chains. So that's Streptococcus. Another one that we will hear quite a bit about when we start to talk about bacteria and health. This here is a very distinctive cell shape. This is a spirillum. And the genus is called spirillum, named for its shape. And here it is here. This often very large cells. And notice this lazy S shape in space. Spirillum very often has a bunch of flagella at the end, here at each end there. When you see it locomoting, it locomotes with these flagella, and it may spiral through the medium slowly, like that. Make a note of that because it becomes important in a minute. These here are curved rods, and this shape is important and very famous because it belongs to a particular genus called Vibrio. Now, there are many curved bacteria, and we say that they are vibrioid. That means they look like Vibrio. But Vibrio is an important genus because there's one species in particular, Vibrio cholerae, which causes diarrheal disease, a very, very violent diarrheal disease. And several of the Vibrios do that. But they have this characteristic curved shape, which we refer to as being vibrioid, curved rods. Now here's the reason that I asked you to remember this lazy S-shaped curve. This is the last of the bacterial cell types that we're going to talk about. This is a spirochete. And the spirochetes are quite distinct from all of the others. First of all, let me explain. They actually have a flagella structure. But the flagella structure is not external. It runs through the center, and you can just see there. You see that kind of line running through here. It's there. And then the cell itself wraps tightly around that flagellum all the way down to form this corkscrew shape. And they have all of the other rods and the spirillum, not the cocci, but the rods and spirillum, they move by these exterior flagella that propel them through the medium. But the spirochetes have a very distinctive, peculiar locomotion, and that is that they spin on their own axis, like this, very, very fast. And they are the only bacterial type which can move forward and back. They can, you distinctively see them doing this, going forward and back against a surface, for example. And the spirochetes are very common in environmental situations. They're also very common commensal bacteria. That is, they exist in our microbiome. They do us no harm, and they probably do us quite a lot of good. For example, you can easily isolate spirochetes if you pick the from between your teeth and mount it. You can do this in lab, by the way. If you want to do so, you just pick a little bit and mount it on a slide in some saline and have a look and see if you can see some spirochetes. They are difficult to see, however, because they're extremely thin. And we usually, to visualize them, use a special kind of microscope lighting called dark field. We actually, the rest of the microscope field is in pitch darkness, but light is shone from the side and bounces off the bacterial cell, and we see them as white against a black background. Makes them easy to see. The reason we're particularly interested in the spirochetes is because there are genera and species of spirochete which cause very serious disease. Lyme disease, for example, is caused by a spirochete. So is the disease syphilis. It's caused by a spirochete called treponema. There's another disease called yaws, which is also caused by, so they can be the cause of very, very serious illness. But there are also many which are completely harmless, environmental spirochetes, and also commensal ones as well. So now remember these: rods, cocky and their different arrangements, the spirilla bacteria, curved, and the spirochetes. I will also tell you that we sometimes refer to the arrangement of rods in the same way. We'll often refer to streptobacilli, for example. Bacilli, which are in long chains, is quite a common description. So cell form, what's its shape, arrangement, how are those individual cells associated with one another. Okay, so I'm just going to finish off today's class by talking very briefly about this incredibly important feature of the cell, which is the cell membrane. At the very start of our microbiology classes, I told you this is the distinguishing feature of the cell, is the cell membrane, the plasma membrane that encloses the cytoplasm of the cell. It is the defining feature of the cell. And the reason that it is so important is because it allows conditions inside the cell to be different to conditions outside. The cell membrane is a living thing. It is continually exerting control. It's controlling what goes out, and it controls what goes in. And it keeps inside things which are useful to the cell, and it accumulates them. So that when we look at the inside of a cell, it looks and functions completely differently to the outside, thanks to the activity of that cell membrane. So it behooves us to understand at least a little bit about how that cell membrane is constructed. So the first thing you have probably seen this before, but the cell membrane, the plasma membrane, consists of two leaflets that are associated with one another, like a sandwich. And here is the one here, and here is the other one here. And these sandwiches are made up actually of an oil, of an oil with a particular structure. This is a phospholipid. These are both phospholipid layers. Lipids are oils. They are substances which are basically strongly hydrophobic. That is, they repel water or are repelled by water. So that is the most amazing thing of all. Enclosing every cell is this thin 2-molecule thick layer. And yet everything, every single function, every piece of integrity that the cell has, and all of its functions depend upon this membrane. That is how important it is. So we'll leave it there for today. And please, if you can, just read ahead a little bit, because it will help you to understand the subsequent things I'm going to tell you, especially about the function, the structure and function of these individual lipid molecules themselves. Okay, and I will see you again.